Hi everyone. You guys have learned already how to enumerate bacterial cultures, how to figure out how many microbes are in a culture. We've just been talking about that in our virtual lab series. And you've learned that you can do this with a spread plate or a pour plate, and that you can do this by making a nice big dilution series of the culture, and then either spread plating or pour plating those diluted samples. There are other things other than sort of E. coli cultures or other cultures that you can also enumerate the number of microbes in. So you will be looking at E. coli in the lab and enumerating a culture of E. coli using spread plates and pour plates. But what we want to show you guys today is how you might want to check in a probiotic sample, so a sample of a capsule or yogurt that contains live bacterial culture, how you might enumerate exactly how much bacterial culture is in there, how many bacterial cells are in there. So we have a couple samples today that you can see on the video. We have some yogurt, some crema there that has live culture in it, or says it has live culture in it. We have another smaller container of yogurt there that has live culture in it. And we also have a kefir, a probiotic drink there in the kind of taller green sort of flask there. And these are all things you can purchase at the grocery store, and they all, when you're eating them, have live bacterial culture. Typically a lot of lactobacillus in it, and that's actually really what we're going to be enumerating with our technique today. There's also over there, at the end there, you can see that there's a capsule or a bottle of a probiotic capsule, so a Natural Factors probiotic that has, it says 19 billion active cells, presumably per capsule. So we're going to start to check that out today and see if it truly has 90 billion active cells per capsule. So what we're going to do first is look at or prepare our samples. So we're going to get ready with dilution, essentially, of these two so that we have a liquid sample that we can then start diluting further in order to make our spread plates. That spread plates, we'll see that in a later video. So we're going to start off right now with actually just getting these guys ready to make our dilution and make and then eventually make our spread plates. So Sabrina's going to step in, and she's going to choose, we're actually going to choose one yogurt sample, and we're going to choose one of the capsules, and we're going to show you guys how exactly you would go about getting ready, because you can't stick a capsule on a spread plate. We need to prepare it before we get going on this. So this is always a little bit of a funny thing because, you obviously eat yogurt at home not using proper aseptic technique, but remember that we're going to be wanting to prevent other unwanted microbes from the environment getting into the samples we prepare, because that's going to confuse our experimental results when we're finally trying to enumerate these cultures. So Sabrina is going to work with the capsule using good aseptic technique. Right now, she is preparing 5 mils. She's going to start preparing 5 mils of distilled water, and we're going to dilute out the contents of that capsule in that distilled water. So she's got a glass pipette right now. She's got a pipette aid that's going to help her pipette up that 5 mils of distilled water. She's going to go ahead and flame the neck of that flask of sterile water that we have prepared. And then she's going to use that pipette to draw up 5 mils exactly. And then we will show you guys how we're going to get the capsule into that as well. So she's just checking the meniscus there to make sure that we have exactly 5 mils. And then it's going to go into a sterile test tube. So it's an empty test tube that has gone through the autoclave, so it's fully sterile inside. So we're just not introducing anything else that could contaminate our sample, so that all we're looking at is the bacteria in that capsule. Notice how she's still, even with that pipette aid there, that green thing that lets her drop the water, she's still holding that capsule, that cap from the test tube in her hand. She did her flame to make sure that the neck of that test tube, the top of that test tube stays nice and sterile. So she's working right within the sterile zone still, even though she's getting out a capsule that obviously came from the grocery store. But once again, you want to just prevent anything getting in there. Now she's just going to flame the neck of that, or the top of that test tube, and she's just going to break open that capsule right down the center. You should be able to kind of just break it open, and we'll just there, if you can, it's hard to see. I can zoom in a little bit and see, but it's, oh, you actually can see the powder from the inside of that capsule, which is all your desiccated bacterial culture sinking into that distilled water down there. Just flaming the neck of that tube again. And now we have that entire capsule's contents are in that tube. She's going to vortex it at this point because we need to get all of that sort of powdery bacterial culture, and you can actually see it starting to look much like a kind of turbid broth culture that you've worked with before. Just gonna, there, yeah, you can really see that nicely. It looks kind of like a thick bacterial culture. So at this point, we're ready with that probiotic capsule sample. So the next steps that you'll see is that being diluted and then going on to spread plates. Right now, Sabrina's going to show you how you would set up the yogurt if you were to use that. Because you want to know, it's no point just adding a spoonful of yogurt in there and saying, well, that's how much yogurt is in, or how many bacteria are in a spoonful. We want something more accurate. So we want to weigh out, in this case, we're going to, you know, pick kind of an arbitrary number of grams of yogurt. We're going to do 2 grams. It's good for creating a small sample. Often you might be using much more than that if you were trying to really get a good feel for the number of bacteria in a sample. But we're going to go with 2 grams. And she's going to use that, I think it's Ioga Activa, looks like looking at the color there, we've got some strawberry action going on. And she's going to start scooping out. So she's got a sterile test tube again. It's a little hard to see there, but you can kind of see it behind the Bunsen burner. Awesome. Yeah, that works great. So she's going to start tapping in. It's always a bit awkward to do. Tapping in some yogurt into that, and she's already zeroed that. So that test tube is sitting in a beaker on the scale. It's being zeroed so that we know, exactly how much yogurt is going on there. She's even got the cap of the tube there as well, because that was part of the weighing. And once she's got 2 grams in there, which it looks like she does, she's now going to basically rinse that yogurt down into the bottom of the tube with 5 mils of distilled water. And that's going to give us roughly, I mean, there's obviously a little extra volume from the amount of yogurt, but it's going to give us roughly 2 grams of yogurt in 5 mils of distilled water sample. It's definitely easier to do this with capsule in terms of getting the whole contents in and not having to awkwardly try to get yogurt off the top of that. But you know, it's always very interesting to look at yogurt, and you guys will get a nice data set at the end of this of sort of yogurt versus a capsule to compare just how much bacterial culture, how many bacterial cells are actually in that 2 grams of yogurt. At this point, she's going to give it a vortex. So there it is, getting a nice tornado there of a vortex so that we have a nice, well-mixed yogurt in 5 mils of distilled water. And then over there at the back, hiding, is our 5 mils of distilled water containing the contents of 1 probiotic capsule. So we have our 2 samples ready to go, and we'll be back with our next video, which is going to show us a dilution series of these 2 samples and then spread plating them on MRS agar. Hi everyone, we're back again now to show you the next steps of this probiotic enumeration lab, where we're going to look at the capsule that we emptied out into 5 mL of distilled water, and we're going to make a dilution series of this, and then spread plate diluted samples of this probiotic on demand rugosa sharp media, or MRS agar. This MRS is selective for lactobacillus species, so it's going to encourage their growth. So we'll be able to see, really enumerate the lactobacillus species that are present in this probiotic. So as you can see, I have that kind of in focus right now. On the whiteboard, we have drawn out our entire dilution series. So anytime you are going to be preparing a dilution series and doing spread plates or pour plates for enumerating bacterial cultures, this is what you would do. You would either write it out in your lab notebook, or if you're lucky enough, as you guys will, when you do this in person in the labs, you'll have a schematic that we've prepared for you. Or in this case, we have a convenient whiteboard, so we spent some time writing it out on the whiteboard so we know exactly what we're doing for all the steps here. So I'm not going to go through it because we've gone through it in class already anyway and gone through similar dilution series. I'm just going to let Sabrina come on in, and we're going to start showing you guys how you do this. So this is going to be, although we're using the probiotic sample right now, when you guys come into the lab, for your labs where you do spread plates and pour plates, you will be looking at E. coli most likely. But the steps of this are exactly the same. So you heard the click probably there of the Bunsen burner going on. So obviously aseptic technique is very important again, as always. So we first set up kind of our workstation. What you can see there is that Sabrina has a lot of tubes. So all those tubes with the red tape labels are our tubes that we're going to use for the dilution series. And you can see there's a lot. You can see on the board as well. We're going to be diluting this all the way down to 10 to the minus 10, because there are a lot of bacteria packed into that capsule. So we really need to make a big dilution, or we're going to have, as discussed in class, that just lawn of bacteria on the plate, right? So you can't see individual colonies and you can't enumerate them. So Sabrina's going to get started by vortexing again. So there's been obviously a break since we last vortexed that. So the last thing you do before you do any transfers in a dilution series, any movement around, is always mix. Because uneven mixing is probably one of the biggest problems for having a dilution series that doesn't give you accurate enumeration. So right now, Sabrina's getting a pipette tip on the micropipetter. She's got it set at this point to 100 microliters. So she's actually going to make an initial dilution of 100 microliters. You can see her flaming the tube there, into 9.9 mils of saline. So as discussed in class, that is a 100-fold dilution, or a 10 to the minus 2 dilution. So she's going into there. She's put in her 100 microliters. She's flaming her tube, discarding her tip into the biohazard there. So there's the tip going in. And the next thing she's going to do, and this is going to get so repetitive, guys, is mix. So she's mixed again, just to make sure there's even distribution of those bacterial cells in that saline solution. Now she's using good aseptic technique. She's transferring another 100 microliters from that culture over into another 9.9 mils of saline. So now, because we were taking from a 10 to the minus 2 already, we've done another 100-fold dilution. We're at 10 to the minus 4. So we do these big dilutions up front because we know from experience that the number of bacteria in that capsule is such that if we just started spread plating bacteria that were only diluted 10 to the minus 2, only 100-fold diluted, we get a lawn of bacteria. So we're not going to start our spread plates and start our our smaller kind of steps in our dilution series until we're around, well, actually until now, when we hit the 10 to the minus 4 dilution part. So at this point, from here on, and you can see Sabrina's readjusting her pipette because she's gonna start making tenfold dilutions at this point. So she's now got she's vortexing that 10 to the minus 4, and she's ready to take 1 mil now from this, 1 mil from that 10 to the minus 4 into 9 mils of saline. And as we discussed, right, when you're doing 1 mil into 9 mils, now you're just making a tenfold dilution. So the next tube she's going to go into will be a final dilution of 10 to the minus 5. And we're going to step down like that, getting more and more diluted to 10 to the minus 6, minus 7, minus 8, etc. So she's made that minus 5 dilution now from her 10 to the minus 4. And once again, there goes the vortex again. She is mixing again, because every single time you go to a new tube in your dilution series, you have to mix what you're taking from first. So here she goes, taking another 1 mil. And this is just going to keep going like this until she's made her whole dilution series. You want to be really sure when you're doing these dilution series that you're keeping really good track of where you are. One of the things that I know myself, often I would make mistakes in, is I would get a little bit distracted, and I'd be like, which one have I taken from? It's very hard to tell. Once you're pipetting 1 mil, you can see the increase in volume, so you can figure out sort of which ones you've added to. But when you're working with the smaller, the 100-fold dilutions, it's hard. And you can see what Sabrina's just done is she's moved some of the tubes that she knows she's already done, and as she goes forward, she'll just put her tube that she's completed, as she's doing right now, she'll put it down 1 row over on the rack. And that is a really useful thing to do when you're working in the lab at any time, but especially when you're doing dilution series, to just have a way of knowing by the placement in that rack that yes, I have added to that tube. And then if you get distracted, you know, coming back to your test tube rack, exactly where you are in your dilution series. I'm getting close there. This is a big dilution series. One of the things where lab work can feel quite repetitive when you have to do it, but of course the end result of figuring out what's in the sample is really nice. So you deal with some of the repetitive moments. And there we go. We have our full dilution series. You can see how Sabrina's really careful to close up those pipette tips as soon as she's done. So those stay closed until you're ready to start working. That pipette box stays when it's open right in the sterile zone, because you're protecting those pipette tips as well, right, from any environmental microbes settling onto them. And they've been sterilized before use so that we're ready to go, and we're making sure we have just bacteria from that probiotic capsule in our sample. All right. Sabrina, do you want to just grab plates, or should we take a break and come back with the spread plates?
Let's take a break, we'll come back.
All right, guys, we'll be back. We've got all our samples ready, and this is actually where you logically could take a break because you've prepared all your dilutions, and then we'll come back, mix those dilutions again, and show you how to make the spread plates. Hi guys, we're back again to finish off this spread plating of our diluted probiotic sample, our natural factors probiotic capsule that we diluted out. So we showed you the big dilution series that we made all the way down to 10 to the minus 10. And now we're going to start preparing the spread plates. So you guys know the theory of this, this idea that if you can, you know, spread out a diluted sample of bacteria so that you get isolated colonies on an agar plate, You can count those, and then you can work backwards using the dilution factor of that sample on that plate all the way back to the original concentration of bacteria in the original sample. And from there, we can figure out how many bacteria were in the capsule. So we're going to start off with that. First of all, we're not going to plate, as I mentioned before, we're not going to actually make spread plates of anything from the 10 to the minus 2 dilution because they're going to be a lawn of bacteria. We know that from previous experience. We're going to start off with our 10 to the minus 4 dilution. And as we also discussed in class, when we spread plate, we add 100 microliters to the plate. So if you add 100 microliters of a 10 to the minus 4 dilution onto that plate, your final dilution factor in that plate, because it's not a full mil, is 10 to the minus 5. So that's going to be, that plate is probably already been labeled by, Sabrina's labeled all the plates already, and it's been labeled with a final dilution factor of 10 to the minus 5, which makes it easy when we want to calculate off of our number of colonies on a plate. So she's pipetting up 100 microliters. She heard the vortex, saw the vortex go, so we vortex that sample, and 100 microliters goes onto that plate. At this point, she's putting it on the little turntable that is a handy way. You don't have to use it, but it's a handy way to start doing your spread. She has taken a spreader, the metal spreader, from a beaker of 70% ethanol, which keeps it nice and clean, and she's flamed off that ethanol. Obviously, if we didn't flame off that ethanol, we would have a problem because bacteria do not like 70% ethanol and they would all die. But a quick flame, we're not holding it in the Bunsen burner to get it hot, we're just flaming off that ethanol. And you can see what Sabrina's doing now is she's testing that spreader. So she's making sure that, well, really cooling it too by touching it to some of the condensation on the lid there. And she is now ready. It's had time to cool because we didn't get it red hot. And she's going to start the spreading process. She goes back and forth all the way across the plate. And as she does that, she turns the turntable. So you can also be your own personal turntable and just rotate the plate as you go back and forth with the spreader. And that works just fine too. Some people find the turntable fiddly and some people really like the little turntable. But you can see she's going back and forth, and you do that for quite a while. One of the things obviously is that you want bacteria spread everywhere on the plate so there aren't clumps of bacteria anywhere. And also you want that 100 microliters to fully absorb into the plate. What she's doing now, which is always what you typically do right at the end, is hold it kind of up against the edge of the plate, as you can see there, and just do the outside rim of the plate. So you're just going to turn, and it's going to go all the way around the rim instead of back and forth. And then once you're done, you want to cover that plate up in a timely manner. You don't want it sitting open to the air. And now you're going to clean off your spreader. So you're going to put it into the 70% ethanol where any bacteria are going to be killed. You do want to be careful always with spreaders that what you'll notice when you're working with them is when you get them into the 70% ethanol and you flame off that spreader, there'll be flames on that spreader as the ethanol burns off. You do not want to, at that point, dunk your spreader into your ethanol because ethanol is flammable and it will light your beaker of ethanol on fire. So don't do that. Usually it can be quickly covered with something in the lab and that puts the fire out. But it's dangerous. So just be really careful that if you do have that spreader lit, or spreader in ethanol and then you're flaming it off, that you wait to make sure that that flame has died down before it goes anywhere near that beaker of ethanol again. So at this point, Sabrina is moving on to the next plate. So now we are working with our 10 to the minus 5 dilution here. And she is now going to put 100 microliters of that onto a spread plate. Spread it in. She's actually going to not use the turntable just to show you how that looks as well. And that plate now, if you remember back to what we talked about in class, that plate is now actually a final dilution when you're calculating back of 10 to the minus 6 because you only put 100 microliters on that plate. She's just testing her spreader before she starts spreading. And then she's going to start the process. One thing you can do, and she just demonstrated that there, just like we talked about with the loops, after you've sterilized your loop, you can touch it to a spot on the plate where there's no bacteria and see if it sizzles. If it sizzles, it's way too hot to start, touching any bacteria with that loop. Same with the spreader. If you touch it lightly to the agar, you should see that it's not sizzling at all. It's not burning into the agar. So now she's doing the hand rotation where she's just moving it with her hand and going back and forth with the spreader. This is actually typically what I do. I think I just worked in labs where we never had the turntable, so we did this if we needed to spread plate bacteria. And now she's finishing off her last little outside rim around the plate, and that looks good. Back into the ethanol for the spreader, on to the next plate.
Just one more.
Yeah, okay, we'll do one more for you guys just so that it's nicely cemented in your head. Remember, you aren't going to be doing this exact lab. When you come into the lab, you're going to be working with a known bacterial sample, just E. coli, in order to practice your spread plate and pour plates. But we'll be actually preparing lots of different plates from a couple of different samples for you guys of these probiotics, either yogurt or capsules, and you'll get a data set so you can start crunching the numbers and figuring out which had the most bacterial cells in it. What was the one that had the most? But we're not going to take you through the full dilution series here, and we're certainly not going to start on the yogurt as well and give you the world's longest video of spread plating. So we'll do one more plate with this capsule sample that we have, the diluted capsule sample, and then we'll call it a day for this video. And like I said, you'll have lots of chance to practice it in the lab, and then we will continue doing some spread plating and some dilutions of some probiotics, and we'll talk about or we'll give you a big data set to play around with to compare some of these different products. Sabrina's just doing her final around the outside with the plate. Lid back on. Into the 70% ethanol, and for now, we're going to close up the pipette. Obviously, you'd go all the way to the end of your dilution series before you would turn things off, but for now, we're going to show you just turning it off. The Bunsen burner is safely off. And usually, actually, in the lab, we just leave the 70% ethanol. You wouldn't dump it down the sink because we can reuse that because it has good antibacterial action, and it can be reused for multiple spread plates. All right, that's all for now. All right, hi again, everyone. We just wanted to show you guys the end steps for the enumerating lactobacilli in probiotic cultures. So we've done all of our spread plates now. So we didn't bore you guys with the whole series of repetitiveness of doing all the spread plates, but they're done now. You can see them in a stack there. Sabrina has them. They are going to go into the incubator for 48 hours at 30 degrees Celsius. You'll notice in your lab manual as you look through that different bacteria require different lengths of time of incubation and different temperatures. And these guys prefer 30 degrees, and they're not super fast growing bacteria, so they're going to need a full 48 hours before we see good countable colonies on our plates. So Sabrina's going to do that nifty trick of tipping that whole stack the correct way. It's upside down, agar side up, but that is the correct way to prevent condensation from dripping onto the surface of agar where you want your colonies to grow. So they're upside down now, and she's going to take them to that 30-degree incubator, and they're going to sit there for 48 hours.
So we're back after 48 hours of incubation, and we can take a look at the results from our cereal dilutions looking at probiotic content. Now, I happen to have that 2 of my samples didn't actually grow any bacteria on them on any of the plates. So that includes our capsule. We can see that all of those plates are totally clear, no growth on there. So no growth at all from the capsule. And when I took a look at that, I did notice that we had actually passed the best before date. So it's quite possible that there were still some active bacteria, but just not enough to show up on that 10 to the minus 5 dilution. The other one that didn't grow anything at all was our kefir drink. You can see again, so this is our 10 to the minus 5 plate. It's the least dilute, so this would have the most bacteria in it, and we really don't see any growth on it at all. The likely reason for that is that kefir actually has multiple different types of microorganisms that are responsible for the fermentation. So it may just be that there wasn't enough microorganisms that liked growing on these MRS plates. Now the ones that actually show us some results that we're going to be able to count and give you data for include our Iogo sample. So here's our 10 to the minus 5 plate, and you can see there that there are lots of little dots on there. I'm going to open up the plate and let you see what that actually looks like. So we can see that there are little dots of bacteria all throughout the plate. And in all likelihood, there are more than 250 colonies on there, and 250 is kind of our countable limit. So we're going to give it a slightly closer look and confirm, but I believe that this one is going to show up as too numerous to count in our data set. Our 10 to the minus 6 dilution, though, again, lots of little dots on there, lots of little colonies growing. Do want to point out that these lactobacillus colonies are quite small when they grow on the MRS agar. But here we've got a good uniform set of colonies growing, and this will definitely be a countable plate, so we'll be able to see some data for this one. The other thing I did want to point out before I move too far away, if we look at this 10 to the minus 5 plate, you can definitely tell that we used good spreading technique because those colonies are all throughout the entire plate and quite even. None of them are clumped together. Then if we look at our 10 to the minus 7 dilution, a little hard to see there, there we go. Again, we've got some colonies on there, and this looks like it might just be considered a countable plate. So anything under about 20 to 25 colonies is not enough colonies to be significant, but this one looks like it will probably still show up on our data set. And then if we look at our 10 to the minus 8 plate, we're really not seeing any growth at all. There might be a couple of colonies off to the one corner. Actually, I think those might just be bubbles, which makes sense. This is 10-fold more dilute than the previous plate, so it, in theory, if that previous plate had about 20 colonies, we would expect this one to have, somewhere around 2 or 3. And it looks like that's exactly what's actually showing up. I can see that I've got 2 or 3 colonies over on this side here. So logically, that would mean that our 10 to the minus 9 plate wouldn't have anything on it at all. If we look at this one, that's exactly the case. It might look like that's a colony, that's actually a bubble in the media. So no colonies on this next plate. Same deal with the 10 to the minus 10 and 10 to the minus 11. So we will have a nice little data set to work with for the Iogo plates. And then if we look at our crema, so that was our Greek yogurt sample, we can see on the 10 to the minus 5, we have, let's just see if you can actually see that well. There we go. So we have a decent number of colonies on there. It looks like this one will probably be a countable plate. So it looks like for this one, we could have actually plated that previous dilution, and we may have actually seen decent growth from that. It probably would have been too numerous to count though. So this will be a plate that we get some data off of. And then our next dilution down, our 10 to the minus 6 dilution, I think I'm seeing one, maybe two very, very small colonies on this one. Not very many though. And that actually does make sense with our dilution series. And then our next dilution, our 10 to the minus 7, we don't show any growth. And we see the rest of that same pattern through the minus 8, minus 9, minus 10, and minus 11. So we're going to take some time and actually count these plates and make the records for them. And that will be data that you're able to see to then work on your assignment for this lab. I just wanted to show you kind of the technique that we're using to count these colonies. So here, this is my Iogo 10 to the minus 6 plate. And you'll notice that over in this quadrant, I've added a bunch of dots with my permanent marker. The reason that I did that is that's how I'm actually going through and counting the colonies. So every time that I see a colony, I make a little dot with my Sharpie as I count it so that I know that I've already counted that one. And so there, we just added an extra 7 to our count, and that brings me up to a total of 87 colonies just in this one little small, kind of about a third of the plate, maybe even a little bit less than that. So since I've already gotten up to that number and I still have so many more colonies left to count, I can already say that this plate is going to be too numerous to count. So we'll just give this guy a T and T C, because it is definitely going to be more than 250 colonies on that plate. And I'm totally okay with that because I also counted the next sample, so the slightly less dilute sample, or slightly more dilute, sorry, sample, and I found that I got 38 colonies on that one. So that is right in the range that I want to be able to use for my data. So I'm quite happy with that and not at all worried that the less dilute sample was too numerous to count. So I'm just going to continue to count these colonies, and I will get you guys a data set that you can work with for your assignment.